My lab has had a long-standing interest in white matter injury throughout the lifespans, ranging from preterm infants who acquire cerebral palsy all the way to the other end of life with patients with vascular dementias, for example, and patients who have multiple sclerosis. So there's a wide number of conditions in which white matter injury impacts neurological function. And we've been trying to understand what are the roadblocks to regeneration and repair. Why is it that after injury to the brain, injury to the white matter, the brain fails to initiate the repair responses that are required for normal function again? There was a key cell type in the white matter called the oligodendrocyte progenitor cell, or what we call the OPC. And those cells we originally showed were very vulnerable to hypoxia, ischemia, and other insults which killed these cells and selectively deleted them from the white matter. So we believed that the loss of these cells was the basis for the failure of the brain to recover from injury. And to our surprise, as we started studying chronic lesions, we found that these cells had a remarkable ability to regenerate themselves and initiate a repair response. There's a molecule in the extracellular matrix called hyaluronic acid. This is a large megadalton molecule. It's one of the largest molecules in the body. It was originally thought to be the glue between cells that holds them together. And over the years, many labs have demonstrated that hyaluronic acid has roles both in inflammation as well as appears to have roles in signaling that have not been more difficult to define. And so we initially demonstrated that hyaluronic acid accumulates in brain lesions in the white matter and blocks the repair process. And indeed, what we found was that the brain lysates would generate a broad range of fragment sizes, ranging roughly from about 150 to 600 kilodaltons. And so this gave us a clue that there was, within that kind of mixture of sizes, a potential bioactive fragment that might be inhibitory to remyelination after injury. We screened those in a tissue culture model system and were able to identify that there was one unique size out of a large range of fragments that actually selectively blocked the maturation of the oligodendrocyte progenitors. We then were able to start defining the molecular pathways that this particular molecule uses to block the repair capacity of the progenitor cells. Now, what was surprising to us when we first tested this molecule was that we looked at a downstream readout of myelination called AKT and expected to find that this hyaluronic acid fragment would shut down the very potent myelination signaling that AKT mediated. And to our surprise, what we found is that initially this hyaluronic acid fragment actually stimulated AKT activation, which was quite paradoxical. And so we started to delve into this finding and realized that what was really going on is that there was a transient activation of AKT and then a chronic inactivation of AKT. Looking at this pathway, we then realized that the roadblock was actually quite profound and that other very potent signals for myelination were not able to detour around this particular repair block. The remainder of the study then traced a molecular pathway which bore a number of similarities to the kind of pathways that have been previously defined for endotoxin tolerance. And we found that, in fact, that there was one single receptor, the toll-like receptor 4, that was activated by the hyaluronic acid fragment and would then signal via an alternative or non-canonical pathway to shut down AKT. Now, what is downstream of AKT? There's a number of potential transcriptional regulators that we considered, and ultimately identified one called FOXO3, which turned out to be a very interesting transcriptional regulator of myelin gene expression, which was specifically controlled by our hyaluronic acid fragment. Interestingly, we found that FOXO3 selectively accumulates in the OPCs that we had originally shown in the baby brain and in the aging brain to accumulate. So there was a very nice smoking gun here where the cells that are blocked in their repair capacity were the only cells that were selectively accumulating this FOXO3 within the nucleus. That raises the interesting opportunity that we might be able to actually use FOXO3 as a readout for, or as a biomarker for studying repair, the repair process and therapies that we might consider to stimulate repair. The tantalizing idea, I think, is that we might be able to stimulate the repair process by blocking the generation of this bioactive hyaluronic acid fragment using more selective and potent hyaluronidase inhibitors than we've had in the past. So that is an exciting direction. We're currently looking at a number of candidate molecules that might be potent hyaluronidase inhibitors. And of course, within the pathway that we've defined, there's a number of other potential branch points where we could think about translation, including at the level of the receptor complex that interacts with hyaluronic acid in the fragment, as well as some of the downstream signaling components. So I think there's a number of exciting opportunities to utilize these new insights to promote and accelerate the progress with regeneration and repair of myelination disorders throughout the lifespan.